Okay, and we're going to turn on guided access. We're going to try on guided access to lock us in. 40 minutes, check that out. And just triple check that, click that home button. And you have options. Let's see. Yep. Okay, intracellular receptors, glucocorticoid. Binding of a blank cortisol to the glucocorticoid receptor. And remember, what kind of receptor is this? This is intracellular. Intracellular receptor. It's is it a catalytic? What kind of receptor is it though? Is it a catalytic receptor? It's a transcription activator. It's transactivator. Dimerizes. That makes it sound catalytic, but I don't think it is. Anyway, it's just its own category of intracellular receptors. Okay. Results in dissociation from heat shock protein 90. Translocation of the GR, dimerization with another GR, and then go to the nucleus to make an activation. Oh yeah. I wonder how it goes to the nucleus though. How does it find its way? Do you know the way? Do you know the way? How does a glucocorticoid receptor know how to make it to the nucleus? I bet the scientists know. I bet they published a paper on it. I bet they were looking at it and they were like, oh, here's the tag, here's a little marker. Here's what picks it up. I wonder if it's transported by anyway, but this is not membrane bound. So that also is what makes me curious, because when you're bound to a membrane, you have these little kinesins to walk you over there, but let's see how they're activated. Yep. Yeah. And of course it's important to notice that it requires another activated. I mean, sometimes the signal is amplified, but in this one it seems like it's maybe because it only requires 2 glucocorticoid receptors in order to activate transcription. Well, it depends on how well directed it is. How does it find its way? It does seem to try to make it as difficult as possible. You have to. It has to get the cortisol has to get through the cell membrane into the cell. Find not 1 but 2 glucocorticoid receptors. They have to find each other, and then they have to find their way to the nucleus. That's a relatively difficult proposition. It's a lot harder than a cell surface signaling. It's a lot less sensitive. So clearly we do a good job of our adrenal cortices do a good job of secreting lots of cortisol. And the glucocorticoid. I should probably fix that. Glucocorticoid. There you go. Intracellular receptors. It is also clear that activation of blank may elicit transcriptional repression as opposed to activation of transcription. Another thing, I should have done this yesterday because, again, I have all the ones from yesterday, so I went from 200 to 358, which is a lot of cards, and that's basically how many I had yesterday. Now it'll go faster because I'm more recently familiar with these guys, but still. Alright, it is also clear that activation of blank may elicit transcriptional repression as opposed to activation of transcription. That would be what are my examples? Steroid hormones, thyroid hormone stimulate, and TSH and I think VDH. I'm not entirely sure. VDR, sorry. Right. Thyroid hormone, or thyroid hormone receptor, right? VDR. See, that's the problem. It's not parallel constriction. It says steroid hormone, and you see a receptor after that. Details, man, details. But what the heck is a VDR? Is it vasopressin? See if I can look it up. No, I'm not allowed in guided access. Okay, well, stay focused now. Don't distract yourself with trying to learn the details. Alright, certain steroid hormones, e.g. ER and PR, estrogen receptor and progesterone receptor, have well-described non-genomic rapid signaling actions via interaction with other signaling mechanisms, like SOC, tyrosine kinases. So we know that at least some steroid hormones are tyrosine kinase associated. Some steroid hormones activate tyrosine kinase associated receptors. The body's sensitive to those steroid hormones because that is the catalytic receptors do present on the cell surface. Moving forward. Receptor and signaling mechanisms selected disease state. This is pseudo-hypoparathyroidism, where the G-alpha-S protein is a defective of the parathyroid hormone receptor, PTH, parathyroid. And yeah. And then there's a pseudo-parathyroid, or hypoparathyroidism, which is a fun one. Alright, hormone/receptor interactions alter target cell function by a variety of mechanisms. Alright, ionotropics. Alright, calcium or sodium into the cell. Natriuretic. Atrionatriuretic peptide NP. Nervous system. Divided into two anatomical components. CNS, PNS, right? Sounds like I'm saying penis. I mean, alright, in the peripheral nervous system, in the penis, sorry, how you get demonetized. The YouTube algorithm also going to think that I said, okay, the PNS, the periphery, alright, in the peripheral nervous system, ganglia, groups of neuronal cell bodies in the peripheral nervous system. Singular ganglion, alright, nerves. Groups of axons traveling together in the blank, and neurons are both present in the ganglia. Glia. And they're all wrapped up in glia. Peripheral nervous system glia. Let's see. Yep. I'm sorry, I'm contemplating my studying. So at some point I have to prepare for my other one. And I think I'm concerned that this information won't have had enough time to sink in. So I'm thinking about subtracting time that I have studying this deck and studying more of the other one earlier, especially considering like how much detailed information there is in the other, in the gastrointestinal. So I think I'm gonna have to start doing that, even if it means not getting through all these cards. The blank brainstem and spinal cord, no Oxford comma, come on, are especially important for the control of the brain. Oh, the hypothalamus. That's it. Hypothalamus, yeah. And we're gonna fix that. We need an Oxford comma up in this. Save, yes. Okay, central nervous system. Spinal cord, integrate. Can process and integrate this information. That's the key word here. Integrate. I'm going to create a brand new card because we need more cards, clearly. And that's going to be integrate. Okay, can process and integrate. All right, medulla oblongata. Responsible for sensing and controlling vital autonomic functions such as breathing, digestion, and blood pressure. Yes, okay, breathing, digestion, and blood pressure. BDBP. Medulla oblongata. That's its introduction. When it comes onto the screen and does this little Power Rangers dance, its slogan is BDBP. Receive sensory afferent information. Oh, it does balance. Okay, that's cool. Breathing, digestion, blood pressure, and then balance. Lots of B's, man. BD. It's like, we'll just call it the triple B. The BBBP. No, BBBD. Ugh, okay. Pons conveys information about movement. Alright, so the medulla oblongata is like, oh yes, breathing, digestion, and blood pressure. But then also balance. And the pons is like, dude, pass your movement signals through me, cerebrum and cerebellum. I'll coordinate y'all. I'll help y'all coordinate. And also urination. Okay, well, which is a motor function. I mean, that's a motor function. Respiration. Right, okay, so medulla oblongata, I suppose, and the pons are responsible for different parts. I imagine that the pons is essential for, I mean, it goes to the medulla oblongata, but the pons is kind of the side. I imagine that there's some, like, processing, maybe that's how the conscious, like, the autonomic and the somatic nervous system integrate to control breathing, somewhere in the pons. Cerebellum. Important for coordination of movement. I don't know why the cerebellum is German. Coordination of movement and posture and balance. Yep. Midbrain controls many sensory and motor functions, including eye movement and the coordination of visual and auditory reflexes. Alright. So when you're midbrain, just like receiving those signals, and it's like, gotta do some reflex stuff. And what does the midbrain consist of again? I think isn't that like the tectum and the tegmentum and the Also, like the substantia nigra and the I don't know, I can't look it up. Alright, it regulates. Can I search for it? Midbrain. Copy. Find. Paste. Midbrain. Nope, that's the only place it is. Okay. Okay, so the pons is like breathing and motor movement coordination between the cerebrum, cerebellum. The medulla oblongata was like, I am important for BBBDP. It's like, no, PDB, BDD, PD, whatever. Breathing, blood pressure, that other B word. Bumbling. And then blood pressure. Let's see. Digestion. Bidirectional communication. Yeah. I expect bidirectionality. Does receive sensory information. A balance, that was the other one. Right, in order for it to control balance, it has to receive signals. So that's a meaningful distinction, right? Like, I can remember that they do different things, but I should also remember that while controlling these facets, the medulla oblongata actually receives sensory information so that it can do its job like coordinating balance. That's why your balance kicks in so quickly and easily, I guess, is because it has this really refined circuit in the medulla oblongata. In the cerebrum, cerebral hemispheres, you have the subcortical nuclei, including basal ganglia, cortex, generation of skilled movements, reasoning, learning, memory, that little thin layer, a little rind that kind of goes across the whole thing. That's your smart part. It can also influence autonomic function. Ironically, it's like, anyway. It's also the worst at its job. Like, all that lower level stuff that's been refined over the course of millennia, and that's the hard part. And this is stuff that everybody can do because everybody's been working on it forever. And we're just got this little tip of the iceberg where we start venturing into like abstract thinking and stuff, and like mathematics and physics, and it's just really not great. Has not had much time for refinement. So we can like build computers that can do that stuff way better than we can really easily. But then it's been difficult to build computers that can do the hard stuff. Anyway, in a sense. Complex behavior, autonomic function. Thalamus processes much of the information reaching the cerebral cortex from it. Okay, moving forward. All right, the central nervous system, CNS, can be divided into 7 main. Yes, okay, we just went through that. All right, sympathetic nervous system, efferents, red. Efferents, that goes away. Do you know the way? Okay. Postganglionic neurons. Our preganglionics are long. Stimulates growth, erythropoietin. That is released by the kidneys. Erythropoietin is released by the kidneys. I'm not sure what cells of the kidney do it, but the kidney does do it. Sorry. Volume. Just remember the volume. 1 liter for saliva, 1.5 to 3 liters for the gastric juices. Oh, here we go. 6.5 to 8 liters, right? Total. Pancreatic secretion is 1 liter. Yeah, I was looking at I should really average out the pHs. But I was looking at what's it called? I was trying this card deck that somebody else came up with and put on Study Blue. And I've never used Study Blue before, but anyway, they put 8 milliliters for how much is secreted, and I think they saw that liters. I'm not sure if they're there's some of the cards that just don't make any sense. Like one was said it secrete, it was saying it was secrete something secreted receptors. Like, you don't secrete receptors. It's that's not how anything works. So clearly the person who was doing this deck needed the help from this class. Anyway, okay. I'm not sure if you're this confused. I'm not sure there's any help for you. And so they said 8 milliliters because they're 8 liters. If only 300 or 100, I think it's pretty sure it was 300, but if only 100 milliliters are lost in the feces, then you can't have 8 liters going into the intestines. And it's yes, you can. Even the large intestine secretes 200 milliliters, which is a lot. Hello. Squeeze. Okay. That's volume. All right. So let's see. So my upper range is small intestine secretes 1.8. That's important. That includes the duodenal secretions, I suppose. And bile is 7.8. Small intestine is 7.8. Large intestine is 7.5 to 8. Centered around the same. They're all kind of basic. Our intestines are a bunch of basic. Super basic. Okay. Values for a normal lean 70 kilogram man. Plasma volume is between 4%, 5% of body weight. Thank you. I'm gonna have to think about this for a moment. So 0.6 is that, right? 0.6 of body weight is total body water, and then 1/3 of that is intracellular fluid. Extracellular fluid, which is 0.2. And then of that, 1/4 is so divided by half, divided by half again, that's 0.5. 0.05. It's about 5%. So 4% to 5% is about right. Yes. Success. Alright, hormone stimulates growth. It's growth hormone. Yes, and that one we've learned is the catalytic receptor. Cannot remember the details. Shoot, that's bad. It's bad if I can't remember the details. I have to remember which hormone goes with which catalytic receptor, and that helps me remember how the catalytic receptors work. Okay, efferent. I definitely know more about growth hormone as a molecule. Efferents. Okay, this is the new 2-neuron chain. Yes, one day. Bronchioles, lungs, right. Chloride, so 1 to 5. 5 times as much extracellularly as internally. 5 times as much in the extracellular, a lot more with the sodium. Sodium's 1 to 10 though, but chloride, it's only like 1 to 5. I wonder if that has to do with the outside cell environment being, the body being basic. Cardiovascular system. Major organs. Heart. Right. Distributes body cells, water, and dissolved minerals, including nutrients. Yes. Whatever. I don't really. Plasma. Why do I have 7 on learn cards now? Hold up. Oh, I see what's going on. It's good, but it's red because I'm going to use it again. Alright, so let's see. Distributes. Did it sound like a cold? Sound like a cold. Okay. Distributes, including nutrients, waste products. Yes. Alright, 4.5 plasma. It's about the same. 4.4, slightly less. Problem is, I don't know if I can remember that off the top of my head. Let's see. Growth hormone stimulates growth. Alright. Released by the kidneys. Capillary wall is the barrier between the plasma and the interstitial fluid. Semi-permeable membrane, where exchange occurs. Capillary wall. Hydrostatic blood pressure at the arterial end of the capillary is higher than the colloidal blood pressure, so the water is forced out into the tissue, and it's called filtration. This gotta be filtered. Interstitium, yes. Gonads, endocrine system, and hypothalamus. Reflection coefficient. Alright, just causes cell swelling. No change in osmolarity because it takes the water with it. Regulates everything. Okay. Renal and urinary system. Red blood cells. Blood ion concentration of the pH, right? Rise, follow, same. Osmotic equilibrium. fall. Definitely fall. Values for normal lean, 70 kilogram man. 4 and 5%. Hypotonic. Yeah. Shifts to the and osmolarity changes in the cells. 70 kilograms, 1.5 liters a day. If I actually drink like 3 liters a day of water, I'm losing about half of that in sweat. So 1.2 liters per minute is filtration. It reabsorbs vast majority of fat. And then filter is 1,700 liters. Wow, that's a lot. Look at that. It's crazy. It's gotta comb through all of it, you know. Can't leave any little bit of water unfiltered. G protein coupled receptors, ionotropic, and catalytic. Yes, enzyme linked. Catalytic enzyme linked. That's right, tyrosine kinase. And then some of them is built in. The ones for the ones that are associated is not. Intracellular, there are 3: vitamin D3, steroid, thyroid, vitamin D3. We talked about those. So there are cell surface. Okay, cool. So these are the 2 main classifications. That's why I was confused earlier. We have cell surface, and then we have intracellular. Okay, hippuric acid. About 0.7 grams of hippuric acid, 0.7 grams per day are excreted through the urine. Wow. I mean, I remember the heck out of that. But, let's see. How do I have more? Anyway, 4.4. 2 lobes, only 2 lobes. Heart's in the way. Hydrostatic pressure into the interstitium. Leak. And ligand voltage aquaporins channels. Passive diffusion, stretch activated. Sodium chloride is impermeable. Yes, but urea effect, no change in osmolarity, but definitely cell swollen. Gallbladder. Pancreas. Oh, look at that. I went down, I got through 100 cards. Look at that, over 100. See, this is a lot faster than yesterday. Alright, just general characteristics, GPCRs. I'm spending a lot of time on receptors. Because I need to. Peptide, glycoprotein receptor, vasopressin, adrenergic, 5 minutes. 5点钟. No, not that. 5分钟. God. 5 o'clock. No, that's not what I meant. Vasopressin. Okay, yeah, adrenergic. So GPCR types. So you have adrenergic. And these are all like surface, right? Cell surface receptors, right? Intracellular ones are the steroid hormone, the thyroid hormone, and the glycoprotein, vitamin D3. Not to mention VDR or whatever the hell that is. So it wouldn't be that. Wait, no. These are all GPCRs, so it wouldn't have been vitamin D3 steroid and VDR. I don't know. That's weird. Okay. Maybe there's another receptor for those guys. Right, and this is the alpha. It's the alpha part of the parathyroid hormone receptor that's effed up. No, calcium receptor. Okay. Adrenergic receptors. Alpha 1, alpha 2, beta 1, 2, and 3. Yes, vasopressin. GPCRs. Oh, subtypes. So these are just all the different things. Super diverse. So adrenergic, muscarinic, peptide glycoprotein, yeah, I mean, this is why I like doing this because in that other study set, the other thing that was wrong was that it was saying that another thing that was wrong. The first part was like acetylcholine receptor, and then the answer should have been muscarinic and nicotinic were the two basic types, but it only said muscarinic. So I think what it did was it looked at the GPCR slide and saw muscarinic and like, oh, this is the receptor, but actually there's another one that's ionotropic that was also mentioned, and that's the nicotinic. And then, but the subtypes are adrenergic receptors and vasopressin. I'm not sure exactly how those are subtypes. Oh, no. What it's saying is that they're subtypes. Like, for example, in the adrenergic receptors, you have alpha and beta adrenergic receptors. Alpha is more for norepinephrine, beta is more for epinephrine, but they do cross-react. Then in those, you have subtypes. Alpha 1, alpha 2, beta 1, beta 2, beta 3. Vasopressin V1, V2. I think there are more slides that talk about these. All right, and the specificity, right? AT1, AT2. Oh, right, this one talks about specificity. I was like, I think there's a slide somewhere that talks about the specificity. Oh, yes, look at this. All right, AT1, AT2, angiotensin 1 and angiotensin 2 are specific for angiotensin 2. Muscarinic, specific for acetylcholine, PTH, parathyroid hormone, but there are more details about specificity. In other words, muscarinics will not bind to antigen. Yes, blah, blah, blah. Yes. Okay. Oh, cool. Wait, it doesn't automatically unlock? Fascinating. Okay, so it'll lock you in, and you only have a specified amount of time to use it. And when it's done, you're locked out. This would be great for like tests, you know? Like if you want to administer a test via iPad, you can do it via guided access. My brain's just always in teacher mode, what can I say? Oh, alright. Yay, all done. Okay, well, that's enough for now. I'm not really all done. Alright, and...
It's break time.
It's break time, alright. I've got to use the restroom, that's